Everyone, thank you for coming. And I am not sure if I'm projecting or will do my best. So it is a real pleasure for me to introduce our speaker today, who is Professor Maura McGrail. She's in the Department of Genetics Development and Cell Biology at Iowa State University. So I will just very briefly give you her scientific lineage, which begins actually at the University of Massachusetts Amherst with an undergraduate degree in biochemistry, which is where her adventures in science began. And then she continued to a graduate program at the University of Minnesota, and there she began her work in the lab with Tom Hayes, which studied cytoskeleton and the role of microtubule-based intracellular transport in development. But really asking quite cell biological questions, but doing so in context of a soft-like embryo. And so that meant using genetic tools which were available in the fly and the fly model. And so by Maura's own admission, that was when the love of genetics began. For her postdoc, Mary joined Larry Goldstein's lab at UCSB, where she continued to use Drosophila to study axonal transport. And then she decided that she wanted to take it up a step or two, the evolutionary ladder. And like so many Drosophilists, like myself included, she became intrigued with the zebrafish embryo for this. I think the main reason was that the promise of the same sophisticated genetic tools that we were enjoying with fly could be brought to a bird embryo. And so I will tell you that this promise is yet to be fully realized. And also that since starting her independent lab at Iowa State University in 2012, Maura has made really important and transformative contributions to making that promise come to fruition, and by contributing to the genetic toolkit that we as zebrafish developmental biologists have at our disposal. So it is really fortunate that we have Maura herself to tell us all about it right now. Welcome.
Thank you, Jenya. So everybody can hear me okay? We're good. Okay. Jenya, thank you so much for that nice introduction and also for the invitation to speak today in the Genetics Colloquium. I am a geneticist, so really it's an honor to be here to tell you about the work that we do in zebrafish to generate novel tools for conditional gene studies. I've had a fantastic day talking with everybody, hearing about exciting research, and talking with graduate students. It's just been a real thrill. So what I want to do today is tell you about how we use CRISPR-Cas genome editing to advance recombinase genetics in the zebrafish model system. At the bottom are my conflicts of interest. I think, as you all know, oops. Okay, I'll do it that way. So as you know, there are many reasons why zebrafish has become one of the premier vertebrate model systems to understand human or model human health and human development. The high fecundity of the embryo allows us to do genetics at scale. In addition, the transparency of the zebrafish embryo really allows us to do things to look at embryonic development and model cellular and disease processes in vivo. And one of the really profound advantages of zebrafish as a model system is the ability to manipulate genes and manipulate protein expression simply by introducing reagents into the single cell embryo by injection. And this, with the advent of genome editing technology, we can now modify the genome in very profound and specific ways simply by introducing constructs and nucleic acids into the single cell embryo. Despite being this fantastic genetic model system, there are certain challenges in zebrafish genetics that we are all aware of. So many of the mutations or genes that we're interested in studying, if we isolate loss of function mutations in those genes, they have essential phenotypes, and this really prohibits understanding the function of these genes in post-embryonic and disease processes. And we also know that many indel mutations or loss of function mutations have pleiotropic phenotypes that complicate our interpretation of the function and role of these genes. And one of the ways that we get around this in the zebrafish model system is to use CRISPR conditional genetics. CRI, it's a recombinase from bacteriophage. It recognizes site-specific sequences called blocks, and it allows for recombination of cassettes. And in zebrafish, we can use this in a tissue-specific way to drive CRI and either invert a cassette that leads to lineage labeling, loss of function mutation, genetic mosaics, and cell autonomous studies. And one of the critical factors impacting our ability to apply CRISPR genetics sort of ubiquitously across the genome or at any gene of interest is, oh, yes, here, sorry, I apologize, is really our methods for generating these tools. Now, traditionally, CRI drivers have been isolated by generating back transgenics that express CRI or a defined promoter with enhancer and other regulatory elements and putting them into a toll-2 transposon and putting them into the genome. There have also been toll-2 enhancer trap screens to identify lines that would have tissue-specific CRI expression. And as we know, with toll-2 transgenesis, integration into the genome is random, and those random integrations can be subject to position effect, ectopic gene extinction, or gene silencing. Now, in contrast to being able to isolate CRI drivers, which has been effective, we have many different CRI drivers from many different screens, many different labs. The ability to actually generate a floxed allele in order to knock it out in a specific cell type or tissue has been very ineffective or inefficient, I should say. And so what I want to tell you about today are our efforts, and this is a resource effort between myself and my collaborators at Iowa State University and the Mayo Clinic to use CRISPR in order to develop robust and efficient methods for generating endogenous CRI drivers and floxed alleles for conditional gene studies. And so first I'm going to tell you about how we developed this strategy for precision integration into the genome, how we validate this approach for CRI-LOX, and I have to tell you some biology about the genes that we chose and why we did what we did in order to demonstrate that we can isolate endogenous CRI drivers and that we can isolate floxed alleles that provide really robust conditional inactivation, and we've also been able to demonstrate this. I'll briefly tell you about some unpublished work. We can use this approach for conditional inactivation of a tumor suppressor to model a form of pediatric brain cancer, an embryonal brain tumor called primitive neuroectodermal tumor. We're very excited about this work. This is a novel way to generate tumor models in zebrafish. And finally, at the end, I'll briefly describe our community CRI resource, where we're generating various CRI drivers and some conditional alleles for the community to really enhance this approach in zebrafish genetics. Now, my colleagues and I, like many in the community, have really harnessed different DNA repair pathways in order to enhance precision genome editing into the zebrafish genome. And as we know, with CRISPR-Cas9, we can generate a double-strand break in the genome very efficiently in zebrafish now. And that double-strand break in the embryo is subject to repair by a number of DNA repair pathways. The predominant pathway in zebrafish embryos is non-homologous end joining, where pieces on either side of the double-strand break come together. And this results in insertions and indels at very high deletions at very high frequency, creating mutations. Homology-directed repair, one of the main pathways is homologous recombination, where we have a DNA template that acts as essentially a template for repair by DNA synthesis. This pathway is essentially has very, very, very low activity in the zebrafish embryo. However, there are other homology-directed repair pathways that, in combination or aggregate, are considered to be microhomology-mediated end joining. And in these repair pathways, if a template is present that has homology, in these repair pathways, homology on either side of the double-strand break comes together to repair the break. Now, this is what myself and my colleagues and others in the field have really tried to exploit. we're trying to harness this microhomology mediated end joining repair pathway to introduce exogenous DNA into the genome at high frequency. And so the idea is that we have our template that we want to put into a location of interest, and we flank that with regions of homology that are complementary to either side of the break. And this is the technology that we have been improving and trying to streamline in Zebrafish. This strategy for introducing cassettes into the genome has been in place for more than 10 years. I just want to briefly point out a few people. The first really work was from Steve Ecker and colleagues, Carl Fleck at the Mayo Clinic, using talons and oligonucleotides to introduce changes into the genome short sequences. There have been a number of labs about 10 years ago who used either long homology flank cassettes or short homology flank cassettes to introduce sequences into the genome with various degrees of efficiency and precision. And so our goal was to really streamline this process of using short homology arms and build a series of vectors that would allow essentially any Zebrafish lab to use this technology to make knock-ins at their gene of interest. The technology that we developed as part of this resource grant, we call GeneWeld, and we are exploiting what was originally designed by Hasano, Yamamoto, and colleagues using short homology to really drive integration. This work was developed by 3 former graduate students in the lab, Wes Wierson, Maya Almeida, and Jordan Welker, who were co-mentored by Jeff Essner and myself at Iowa State. And they're really responsible for the development of this entire GeneWeld system. The idea is actually quite simple. We are interested in integrating a cassette at this CRISPR guide RNA genomic target site. We have a donor vector that has a cargo that we'd like to introduce. We flank that cargo with homology arms that are complementary to sequences on either side of the genomic cut site, and these are on the order of 24 to 48 base pairs. What we did was to put another guide RNA site flanking the entire cassette, and we call this a universal guide RNA site. These sequences aren't present in any vertebrate genome, and the idea is that if we can introduce, when we inject into the embryo, Cas9 RNA, our genomic guide RNA, a universal guide RNA in the donor, the universal guide RNA will liberate this cassette, creating a linear template of DNA flanked by homology. And this happens simultaneously when the cut is occurring in the genome. And what we know from what we published is that this drives integration at very high frequency through the germline, precise integration. And so when we say germline integration, what we mean is that on either side of the cargo insertion site, there are no indels. It's seamless and scarless. Now, the efficiency of this approach using a 2A MRFP cargo was on the order of 22 to 100% of founders that were raised to adulthood would transmit this precise allele. And so we targeted genes that represent, a variety of types of genes. They're expressed at different stages of development. They encode different types of proteins. So as far as we can tell from this work, there's tyrosinase and an endothelial specific adhesion molecule and a transcription factor node 1 and a connexin gene. They don't appear to be impacted by the chromatin around them, as far as we know in this case, right? There's no evidence that that has an impact on the efficiency of recovering these integrations. As part of this work, we developed a highly detailed protocol. We published it in Bioprotocol. And we also have a suite of vectors that can be used with this GeneWeld approach for integration. So we've got vectors with different reporters. If you just want to make a 2A GFP or an RFP, or you want to integrate Gal4, these are available at AbGene. We also have another suite of vectors called PRISM, and this is for precise integration with secondary marker. And these cassettes have, in addition to the primary thing that we want to integrate into the genome, a linked secondary marker with a tissue-specific promoter that drives expression of a reporter, lens GFP, heart GFP, hatching enzyme. So we've shipped these reagents out to more than 70 labs across the globe, and this is really designed to be a resource. So if there's something up here that you are interested in, please don't hesitate to contact me. This is supposed to be supporting the community. And we provide one-on-one Zoom consultation for our protocol. If you're interested in finding out more of what we do, I do this all the time with people around institutions around the globe. So this was a really nice demonstration of one way that we can use CRISPR to make precise knock-in alleles at genes of interest in the zebrafish genome. what I want to spend most of the rest of my time talking about is how we actually use this approach to generate robust CRISPR conditional alleles in genetic tools. And in thinking about how we wanted to do this, use GeneWeld for this, we needed to really think about three considerations. One is we wanted to generate endogenous CRE drivers. So we would take our approach to integrate CRE into a gene of interest that would allow for cell type specific expression under the endogenous gene regulatory elements, hopefully reducing this potential for gene silencing or off-target ectopic expression. The other thing that we wanted to do was generate an approach that would allow us to make true floxed conditional alleles. We wanted to simplify this approach. We need a single guide RNA site that we can integrate a cassette into. It really was imperative that we had robust and consistent knockdown of transcription of these conditional alleles in order to generate robust loss of function phenotypes. And then we wanted to have a fluorescent marker to label knockdown in mutant cells. And then finally, in thinking about the genes that we were going to target to make these floxed alleles and show that the system worked, we really wanted to think about using genes where we knew that these genes were essential. If there was an indel mutation, it had a robust loss of function phenotype only after simple indel mutation. So there was no evidence of genetic compensation. So there's a clear phenotype to follow. We wanted to be able to use these to determine if there was in fact a cell type specific requirement for these genes as their cell autonomy. And ideally, these genes would be required for development, but also be implicated in disease. So if we generated these floxed alleles, could we show that they function as expected for genetic mosaics in the embryo, and could it also be used to model disease? So I want to take just a minute to tell you about the biological questions that my lab is interested in order to provide a framework to explain why we chose the genes we chose to generate CRE drivers and floxed alleles. So my lab is primarily interested in the regulatory mechanisms that control the neural progenitor cell cycle. And we study this in the context of brain development and pediatric brain cancer. And it's clear that in human brain development, the most profound or rapid stage of development occurs from the embryonic to the fetal period. And after that, brain growth slows down dramatically. And so in order to think about how brain development occurs properly, there must be some control of the ability of cells to proliferate and then undergo this process of generating neurons of a specific cell type through specification and differentiation. And in order to have this happen normally during these rapid phases of brain growth, there must be some proper control of neural progenitors moving through the cell cycle, passing that point, entering S phase, and eventually exiting the cell cycle and undergoing specification and differentiation. There must be some really precise control of that process. Why is this of interest? Well, in pediatric brain cancer, such as embryonal brain tumors, many of the tumors are highly proliferative, malignant, there are no effective treatments, and these embryonal tumors fall into a class in general called primitive neuroectodermal or embryonal tumors. There are some famous ones like medulloblastoma and retinoblastoma. There are others that are less well studied, just as malignant. They have many cases across the globe, and as I mentioned, the treatments are ineffective. What we know about these classes of embryonal brain tumors is that these solid tumors are very distinct from adult tumors in their phenotype, in their molecular nature, and their pathology. So there is some limited molecular understanding. Most of these tumors that fall into this class either lack the tumor suppressor RB or P53, either through silencing or disruption of the pathways that control the activity of these genes. They have an overall low mutation load. There's some evidence, there's copy number variation that corresponds to oncogenesis in this class of brain tumors. And there are neural progenitor and epigenetic signatures in the gene expression data that is available, indicating that transcription factors that drive neural progenitor proliferation are actually elevated, as well as epigenetic or chromatin regulators. And the idea behind these embryonal tumors is that it's really a neural progenitor developmental mechanism that is being hijacked. and maintained that allows for persistent growth and proliferation. And one of the major questions is why these developmental mechanisms become oncogenic. What is it that's happening that progresses from simple proliferation and dysplasia to oncogenesis? And really understanding the molecular mechanisms that underlie these tumors, hopefully the idea is that we could identify some tumor vulnerability that is unique to pediatric brain tumors. So we modeled this in my lab by simply injecting into a single cell embryo, either CRISPRs or TALENs, to knock out the RB1 tumor suppressor in somatic tissue. And what we found, and this is all published, I'm just going to briefly talk about it, is that at about 80% frequency, adults will eventually develop these tumors in the brain. They extend along the entire, from the forebrain to the hindbrain. And the pathology of these tumors indicates they are poorly differentiated, highly proliferative, and their molecular signature based on transcriptome analysis is that they represent a subtype of primitive neural ectodermal tumor in which the tumors express the transcription factors OLIG2, proneural, and SOX10+, as well as ASC1B. What we noticed in the transcriptome of our tumor model is that there were a number of genes that were elevated that are part of repressive chromatin complexes like the MURD, histone deacetylase complex, and that includes these genes, histone deacetylase 1 and RBP4, excuse me. And there are other chromatin regulators like histone demethylase is elevated as well. And a colleague of mine at the German Cancer Research Center, Marcel Kuhl, has a database of over 400 glioma tumors in humans. And they have expression data on these tumors. And this is just plots of these different classes of embryonal brain tumors. And you can see that in both cases, both histone deacetylase 1 and RBP4 are highly expressed relative to normal brain tissue. And so our tumor model in zebrafish is clearly mimicking the pathology and molecular signature of these malignant pediatric embryonal brain cancers. Why is this of interest in thinking about RB? So we know that the canonical role of RB to suppress entry into the cell cycle is by facilitating repression of E2F target genes in combination with the NURD histone deacetylase 1 complex. And this adapter protein RBBP4 mediates the interaction of this complex with chromatin. It was originally identified as an RB binding protein. And so our tumor model where we knock out RB shows elevated activity of these, suggesting that in the absence of RB, there must be some function of these complexes, of this complex that is oncogenic, which may be separate from its role in normally suppressing cell cycle entry. So all of this interaction of RB with these complexes, whether or not this actually occurs in vivo in neural progenitors, wasn't really known at the time when we started studying them. So we were interested in trying to gain some insight into the interaction of these genes and oncogenesis by studying them in normal brain development. So we used CRISPRs to isolate mutations in each of these genes, and this is published. Histone deacetylase 1 has been studied extensively in zebrafish. Paul Henyon's group first isolated mutations in this gene called Colgate's. And what we found was similar to what they found. Loss of function of histone deacetylase 1 during embryonic neurogenesis results in a failure to maintain their proliferative progenitor pool. So there's microcephaly, microphthalmia, and there were other phenotypes associated with the vascular development, et cetera. Now, loss of RB1, a simple mutation in RB1, the phenotype is that progenitors appear to fail to exit the cell cycle, or they re-enter the cell cycle. They condense their chromatin, they appear to arrest in M phase for a while, and it isn't really clear what happens after that. Now, in contrast, this adapter protein that's supposed to mediate the interaction of all of these also shows a phenotype similar to RB1 in the developing brain and retina, but eventually those cells undergo apoptosis. So what we have from basic indel analysis of these genes that are supposed to be functioning together are three somewhat distinct phenotypes. So we decided that these genes made a lot of sense to use to validate our approach for CRILOX conditional genetics. We had essential genes, simple indel mutations, strong loss of function phenotype. We could use conditional alleles to ask, do they really interact together, specifically in neural progenitors eventually, once we have those CRILOX alleles. They're clearly required for brain development, and they also play a role in embryonal brain cancer. So this is what we decided to do. We decided to integrate CRE into the genes that encode ASC like 1B and OLIG2, these proneural progenitor transcription factors. We made a CRE driver in NeuroD1 as a control. This is a gene that's downregulated in our tumor model. And then we decided to isolate floxed alleles in these 3 genes. So in order to start this project, the first thing that we did was to use our gene weld strategy to integrate CRE. We've also started working on CRE-ERT2 into the coding region of these genes of interest. And this was work started by Maura Almeida and was finished off with help from Sahiba Siddiqui, who's currently a 4th-year genetics student in the lab, graduate student, excuse me. And the idea is that we would have a cassette that has a linked secondary marker, lens GFP, and we integrated early in the coding because at the time we wanted to make sure this cassette would be under the control of the regulatory elements that would drive expression of our gene of interest. Now the cassette is going to be disruptive, it's going to make a mutation. And so after expression, you would only get expression of CRE, but not functional protein. And so we did that. Myra did this. She isolated these 3 CRE drivers, and the frequency at which she recovered these was similar to what we had found with our original gene weld approach. She was actually able to inject into a switch reporter and raise up the animals that had switching activity. So now we have 3 different CRE drivers that define these stem progenitor populations in the embryo and then mature or neurons that have committed neurons. So I just want to show you one piece of data in how we characterize these CRE drivers to demonstrate that they had activity and expression that was under the control of the endogenous gene regulatory elements where we had integrated them into the genome. So this is the ASC like 1B 2A CRE line. And you can see by in situ hybridization, I believe this is 3 days, that ASC like 1B is expressed in the proliferative zones of the hindbrain and the midbrain and the forebrain. It doesn't show up here, but it is. It's not expressed in the retina, not expressed in the cerebellum, and CRE itself shows the exact same pattern of expression as we expected. And when we use this CRE line in combination with a UV switch reporter, where we've got a flox GFP in the presence of CRE, GFP gets recombined out and the expression switches to M-cherry. All of the cells that are derived from the lineage of ASC like 1B, everything in the midbrain and the hindbrain, this is olfactory, now switch from green to red. There's no switching in the retina where ASC like 1B is not expressed. So we have these 3 lines now that provide tissue-specific or cell type-specific CRE activity. And the next thing that we wanted to do was generate our floxed conditional alleles. And so this work we published last year in eLife, and this again was originally started by Maura Almeida and Jordan Welker, former graduate students in the lab, and really was brought to completion by Dr. Shekhar Kambakam, who's a senior scientist in the lab. He now works at USDA. Zhitao Ming, who's a technician in the lab, has been working in the lab for 6 years. He was 3 years as an undergrad, now 3 years as a technician, as a true gene weld wizard. And Dr. Fang Liu, who joined the lab about 3 years ago. And really these people were instrumental in bringing this to completion. So our goal was to generate flox alleles that would allow for conditional inactivation in specific cell types, and they needed to provide a marker of fluorescence so we could mark mutant cells, and we needed to be able to demonstrate that these genes could undergo conditional rescue in the presence of CRE. In order to build these alleles, what we did was to use a variation on our gene weld approach. And so this construct that we called UFlip has a number of elements in it, and I'll describe them. And really our goal was to demonstrate that every single element that we designed within this construct worked the way we thought it should. And so we call this a universal UFlip vector because it has universal guide RNA sequences flanking it. In theory, you should be able to integrate it into a single intronic guide RNA site in any gene of interest and create a conditional allele. It's basically a gene trap cassette. So we have a splice acceptor followed by 2A mRFP and transcriptional terminator that would provide RFP expression and splice after splicing into it. And we have our linked secondary marker. The entire thing is flanked by alternating pairs of LOXP sites. And this has been used for decades in order to provide stable inversion of a cassette after exposure to CRE. So there's one recombination event and then a second brief one to lock the cassette into place after recombination. One of the key elements within this construct is this transcriptional terminator, because in order to demonstrate robust gene knockdown, we need a transcriptional terminator that will stop gene expression to a very low level, like 1% of wild type levels. And I'll show you the data that illustrates that this cassette can do that. We also have a pair of ROX sites that flank the cassette, and these are responsive to the DRE recombinase, and I'm going to come back to why they turned out to be incredibly useful. So we have two ways that we can integrate this cassette into the genome to create conditional alleles. The first way is just to put it in what we call the gene off orientation. And what happens during transcription is that the primary transcript will splice into the cassette. There's transcriptional termination, MRFP expression, early termination, and induction or creation of a mutant phenotype. The other way that we can integrate the cassette using GeneWeld is to put it in what we call the gene on or passive orientation. So the entire cassette is integrated in the opposite orientation. It should have no impact on gene expression. When the primary transcript is processed, the entire thing will get spliced out. So we did this. We isolated both gene on and gene off alleles for these three genes of interest by integrating cassettes in intron 4 of RBBP4, intron 5 of RB1, intron 6 of histone deacetylase 1. And for the next bit, I just want to use the alleles that we isolated in this histone adapter protein in order to illustrate how the alleles work, because the loss of function of RBBP4 is very robust at the morphological level, at the cellular level, and it allowed us to demonstrate how these alleles can work for conditional inactivation and rescue. So here's the first allele that, the first RBBP4 allele that we isolated. So this is in the gene off orientation. I've simplified the cassette just to illustrate how when we put this in an intron, it's going to cause premature termination, loss of RBBP4 expression, and induction of RFP. And so this is a stable transgenic where we can see RFP expression throughout the nervous system, which we know RBBP4 is expressed there. It's also expressed within the retina. And so the lens, although it expresses BFP, it appears magenta because of the overlap of RFP and BFP. And importantly, when we take this mutation and we put it in combination with a simple indel mutation in this gene, this results in the neurogenic phenotype that we expect from loss of function, microphthalmia and microcephaly. So there's robust knockdown of gene expression as we hoped. The next thing that we wanted to do was to illustrate the opposite with an on allele. And I wanted to just take a diversion and explain how we were able to isolate the opposite allele with very high efficiency using these rock sites that flank our cassette. So we have some evidence that pre-selecting embryos that express our primary reporter after gene targeting, raising those to adulthood, and then screening for germline transmission helps increase the frequency of recovering precise alleles. So if we start with this allele, a founder that has a precise integration, we take embryos from that founder and we inject DRE mRNA, we know that we can flip this cassette at ROX sites. As long as DRE is available, the cassette is going to continue to invert. But once DRE runs out, the cassette will stop in one orientation, the original one or the flipped one. So 50% of the time, you would predict you could recover this inverted cassette, the gene on orientation. So Ming did that. He injected DRE. He only raised 11 adults to adulthood. He screened for transmission of alleles that had lens BFP, our secondary marker, but had lost RFP. And 7 out of 11 adults transmitted this inverted U-flip allele. Incredibly efficient, way more efficient than integrating the GeneWeld cassette and screening 3 to 30 founders for recovering an allele. So now we have our gene on and our gene off allele. So the gene on allele, where the cassette is in the passive orientation, RBBP4 appears to be expressed at wild type levels. This is the gene on allele. There's lens BFP, the marker for this allele. When we take this and we put it in combination, transheterozygous combination with an indel mutation, there's no morphological phenotype. So this suggests that the integration of this cassette allows for wild type levels of gene expression. So now we have these two alleles that allows us to test for conditional inactivation and rescue. The first thing that we did was determine at the cellular level that the phenotype induced by this off allele was in fact due to loss of function RBBP4. So in the indel homozygous mutant phenotype of RBBP4, we know that this neurogenic phenotype is the result of extensive apoptosis throughout the midbrain and the retina at 2 days post-fertilization. When we look at embryos at 2 days post-fertilization that contain the off allele and the indel mutation, we see the exact same thing, extensive apoptosis throughout the midbrain and the retina. And when we look by RT-qPCR at the level of expression of endogenous RBBP4, it's very clear that in homozygous mutants for the off mutation, there is a reduction to less than 1% of wild type levels of expression, demonstrating that robust gene knockdown leads to indels and loss of function, the loss of function phenotype. So in contrast, our on allele that is predicted not to disrupt RBB activity, morphologically, it appears that there's no impact on development or gene expression. The embryos appear normal. There's no neurogenic phenotype. When we look at 2 days post-fertilization, there's actually no cast space that we can measure to any significant degree. But what was interesting and actually very revealing to us is that when we did the RT quantitative PCR to measure the level of expression of the wild type RBB4 messenger RNA, it was clear that this allele has some impact on messenger RNA levels. In heterozygous, there's about a 25% reduction in the expression level of the endogenous gene, and in homozygous, it's reduced to 50%. So for a haploinsufficient locus, this approach might not be effective. But for many genes, we know that in order to get a robust loss of function phenotype, we need to reduce expression to well below 1%. And that's what these alleles allow us to do. Okay, great. So the first thing that we wanted to do, or the next thing that we wanted to do, is actually demonstrate that these alleles would respond to Cre activity and function the way we hoped that they would. And so the first thing that we wanted to do was demonstrate that we could conditionally inactivate a gene simply by introducing a ubiquitous form of Cre. So we start with our passive allele where the gene is expressed. We introduce Cre by injection into the single cell embryo. And then we look for inversion of the cassette. And so we start with embryos that without any injection would be transheterozygous on over delta 4. When they're injected with Cre, what we see is induction of the neurogenic phenotype, induction of RFP expression, meaning the cassette has flipped as expected, and an induction of apoptosis, which is indicative of the phenotype as well. And we quantified the amount or degree of inversion by Cre after injection, and more than 80% of the cassettes in the genome had been inverted by Cre recombinase. So very robust induction of a mutagenic phenotype after Cre recombination. So this is ubiquitous Cre, and I think really one of the most powerful things is to be able to demonstrate that we can induce conditional inactivation in a cell type specific way. And the first thing that we did was to use our ASC-like1B2A Cre driver in order to see if we could induce loss of function specifically in that midbrain neural progenitor population. So we start with our passive on allele. We introduce Cre by crossing in Cre. We also have embryos that have the delta 4 mutation, so it's on over delta 4 transheterozygotes in the presence of this neural progenitor Cre. And what you can see in the midbrain, at high mag over here, is that the cast space shows that there is in fact an induction of apoptosis. And what was important about this experiment, all of the red shows where Cre is expressed, is that there's no induction of apoptosis in the retina. We know that RBBP4 is required in the retina for survival, but there's no induction of apoptosis because this Cre driver is not expressed in the retina. It's only expressed in the proliferative zones of the forebrain, midbrain, and hindbrain. So we had really nice tissue-specific knockout. The next thing that we wanted to do is demonstrate the flip side or complementary experiments where we could demonstrate that we can conditionally rescue a loss of function U-flip allele by adding in Cre. And this worked really well with ubiquitous Cre. So we start with embryos that are trans-heterozygous for the off mutation and the indel mutation, and normally they would have apoptosis throughout the midbrain and retina, resulting in this strong neurogenic phenotype. When we inject CRE into the embryo, this completely rescues apoptosis, rescues the morphological phenotype, and what we found is, again, more than 80% of the cassettes had flipped in the genome, and this was enough to, at least visually, completely rescue the loss of function phenotype, right? The last thing that we wanted to do was the trickier experiment, and like in retrospect, this wasn't ideal, but we did it. So we wanted to be able to demonstrate cell type conditional rescue. Difficult to do when you start with an embryo in which all of this apoptosis has occurred throughout the retina in the midbrain. And so this is just loss of RBBP4, hitting more than one neural progenitor population. We tried to rescue just by inverting the cassette with our ASC like 1B CRE. So a subpopulation of neural progenitors would express CRE and invert the cassette. Visually, it looks like there may be some reduction in apoptosis in the midbrain, certainly not in the retina, but ASC like 1B isn't expressed in the retina. When we look at high mag, there's still cell death. It's hard to see in the blue, but there are lots of cells undergoing pignosis. And there is a slight reduction, but it's not statistically significant in the level of caspase. So it wasn't a perfect experiment, but it certainly suggested that indeed, if we had a cell type specific form of CRE and we had the right condition, we should be able to demonstrate conditional rescue of a loss of function. Very powerful approach for cell autonomous studies and mosaic analysis. So just to summarize, what we've done is to isolate our endogenous CRE drivers. They function as expected for cell type specific CRE recombinase activity. We're able to generate these endogenous floxed alleles with effective gene knockdown, induction of loss of function phenotype, labeling of the mutant cell population, CRE-mediated inactivation and rescue is possible. And we can do this with cell type specific CRE drivers. So this was really exciting for us. And I just wanted to briefly talk about some work that is not published that we have ongoing in the lab to use this approach to model embryonal brain cancer. And in our embryos that we generate from animals that contain a loss of function mutation in the RB1 tumor suppressor, or heterozygous for loss of function mutation in RB1, and have this ASC like 1B CRE driver, when we cross these animals to adults that have our RB1 conditional on allele, so the gene is expressed, we generate animals, or embryos, excuse me, in which the RB1 on cassette gets flipped and locked into place by CRE. And this leads to induction of RFP expression throughout the nervous system where RB1 is normally expressed. So we're essentially generating RB1 on to off conditional inactivation in this neural progenitor pool. And what we found is that these adults, when they become on the order of 5 months of age or so, they form, or tumors form within the brains of these adults that mimic the tumor type that we saw previously just by simply knocking out RB1 with CRISPR. So we've done pathology and RNA-seq on these. So the morphology or the pathology is highly proliferative, poorly differentiated. This is preliminary results. Our RNA-seq indicates that these tumors indeed show olig-2 SOX10, a neural progenitor signature. We see elevated expression of the same epigenetic regulators that we found before. And so this suggests that at least one population in the embryo in which we can mutate RB1 or lose RB1 will give rise to these poorly differentiated tumors. All we have right now at this moment is RNA-seq analysis. And I have a collaborator at Iowa State who's a computational biologist and bioinformaticist. And he very generously was able to take our RNA-seq data and look just within that data, which I know is a very small percentage of the genome, but it's 5' UTR coding, 3' UTR, and compare the rate of indel or SNP between control tissue and tumor tissue. And it was very clear from that analysis that there wasn't any significant difference in the number of insertions, deletions, or SNPs that happened during the induction and formation of these highly proliferative, undifferentiated tumors. And this result is really consistent with this idea that what drives these embryonal type tumors is some modification of the chromatin landscape driven by epigenetic regulation that may underlie this process of oncogenesis. This is speculative and this is preliminary, but what we really hope to do is to use this tumor model in order to understand what is the molecular time course of oncogenesis. This isn't easy to do in other model systems, but we can do it in zebrafish. We can label our simple RB1 mutant cells with RFP. We can capture those cells. We can do ataxia. We can look at epigenetic histone marks, profile those, the entire omics level of analysis. We can do transcriptome and correlate how these change over time as these simple RB1 mutant neural progenitors progress from a simple loss of function mutation to truly transformed to oncogenic. And so this is work that we hope to pursue over the next few years. So the very last thing I want to talk about to end my talk is our community resource, and I hope that this will be of interest to many of you in the audience. What we have done as part of our resource grant from the Office of Research Infrastructure Programs is to build a set of CRE drivers that would mark different lineages in the zebrafish embryo that we hope will be of interest to the community. So in this work, we are generating either CRE and CRE ERT2 with linked reporters, endogenous lines where we integrate this cassette at the 3' end of the coding of our gene of interest. And the idea is that our gene of interest is expressed at normal levels, so it's non-disruptive. And we also have expression of CRE from this 2A sequence. So we've isolated lines in mesodermal and macrophage lineages and endothelial. We're in the process of isolating hematopoietic drivers. I'll tell you some of the work in characterizing a couple of these lines. And I've been really fortunate to have a network of users in the zebrafish community who've been brought on board that are willing to help us characterize these lines. And these are experts in the field for whom these lines would be of use. These are the genes that we have targeted so far. So to label mesodermal lineages, we've integrated CRE and CRE ERT2 and the HAN2 transcription factor. We have generated a CRE driver in the macrophage lineage marker MPEG 1.1. CRE ERT2 is in progress. We have these neural progenitor lines I told you about. We have a FLE1B CRE driver that labels endothelium, and we're in the process of isolating ERT2, CRE ERT2 drivers for these. And we are in the process of isolating drivers in SOX32 for mesendoderm, and these hematopoietic stem cell markers or hematopoietic lineage RUNX1 and GATA2B. And just as one example to show you how they work, and this is HAN2 CRE, and this is as I said, a marker that is expressed in the lateral plate mesoderm and labels all mesodermal lineages in the embryo or most. And the characterization of this line, it was isolated by Ming in my lab, and it's been characterized by Rob Lalonde and Hannah Moran and Christian Mosselman's lab at UC Denver and Chunwei Yin at Cincinnati Children's Hospital. And what their analysis has shown really beautifully is that CRE leads to switching of different reporters. It labels the heart, pectoral fin. This is a ventral view of the heart labeled after switching in the pharyngeal arches as we expected. And here in the liver in a 5-day post-fertilization larvae, there appears to be labeling of both the hepatocytes and hepatic stellate cells. We also have a CRE ERT2 line, and what Rob and Hannah did in Christian Mosselman's lab was to use 4-hydroxytamoxifen to show we take an embryo at the shield stage, treat with tamoxifen, wait an image at 24 hours. What you can see is that there is induction of switching of the CRE responsive reporter. So we can see GFP expression throughout the expected mesodermal lineages. And really importantly, in the absence of 4-hydroxytamoxifen, there's no switching. So we have the ability to very carefully regulate the activity of CRE using 4-OH tamoxifen. This has been widely done, but here we're doing it with an endogenous CRE ERT2 line that should allow, hopefully, for spatial and temporal control at other stages. And as I mentioned, I've been really incredibly fortunate to work with these individuals Ben Liao at Academia Sinica in Taiwan, Diana Mitchell at Idaho, and Celia Shao at UNC, and helping to characterize our MPEG Cree lines. And then Christian Mosselman in Denver, Chinue Yin in Cincinnati, and Jeff Mum also has our HAM2. I'm sorry, he has our MPEG lines. And Chinue and Christian have been working on the HAM2 lines. We've distributed these things around the globe, our vectors, our protocols. We're starting to send out our lines as more and more come on. So I hope that you won't hesitate to contact me if there's a line that you would like to see on this table that would be a benefit to the zebrafish community. So just to summarize, we've demonstrated we've been able to generate really strongly controlled, robust, and conditional inactivation and conditional rescue alleles. We have a novel model of primitive neuroectodermal tumor, which I think provides a new paradigm for generating tumor models in zebrafish and potentially other disease models. And we continue to build on our zebrafish community Cree resource. And so I need to end just by thanking all of the people in the lab at Iowa State. Here we are. In Jeff Esther in my lab, this is Dr. Fong Liu, Zhitao Ming, Dr. Shekhar Kamakam, and our former members, Myra and Shekhar Skano, Stacy, Laura, Jordan West, Melanie, and Trevor, who are really instrumental in building this gene weld resource for CRISPR precision targeted integration. Many collaborators at Iowa State, the Mayo Clinic, Steve Ecker and Carl Clark are our collaborators on our resource grant. Had the good fortune to collaborate with Jenya on some really exciting work using genome editing to study neuroprogenitor cell cycle control, and many collaborators around the country. And our funding from the NIH and the Roy J. Carver Charitable Trust is a foundation in Iowa that gave us money to build our state-of-the-art aquaneering zebrafish facility. And so we are very grateful to them. So I will stop there, and I would be happy to answer any questions. Thank you. Yes.
Something I didn't understand right at the end there, and I was hoping you could expand a little on is like what you mean by the switch reporter.
Yeah, sorry. So this is a heat shock switch line. So there's a heat shock promoter, and that's followed by a cassette that has a lock site, a transcriptional terminator, and a lock site. And those lock sites are oriented head to tail. So after recombination by Cree, the transcriptional terminator is recombined out. And so what ends up happening is that GFP is expressed. So this is a GFP switch reporter. So in the absence of Cree activity or tamoxifen, transcription will terminate at the LOXP site. So the heat shock promoter is leaky, but in the absence of heat shock, there shouldn't be any induction of gene expression as well.
Okay, so thank you. Great.
Yes, Michael.
Oh, nice.
Yeah, you.
Fantastic.
Thank you.
I'm curious with your pre-drivers that you're generating these promoters. These are all using the endogenous promoter sequence. My question is, how do you go about selecting the genomic CRISPR site that you're targeting, and how much effect does that have with how well that Cree line works? And I guess another concern is, oftentimes you find these regulatory regions that are tens of megabases away, or within introns and all of that. So have you found any problems with this so far? Or there anything that won't work, or what do you go about in selecting something like that?
So, great question. So for all of these genes, what we have done, just in case something doesn't work, we always target at 2 locations early in the coding sequence and at the 3' end right before the translation termination codon. And what we have found for everything except MPEG 1.1 Cree, they all work. And so full disclosure, we have an MPEG 1.1 Cree line where we integrated it at the 3' end before the translation termination. It doesn't work. We don't know why. No idea. I wish I knew. I'm glad that the genome still holds mystery for us. We haven't figured it all out, And so, and that was really a surprise to us because everything else worked. You know, all of the lines that Maura, the first one she made, they all worked. You know, she picked a sequence in the coding region, it worked. And, you know, so I guess there's still some trial and error. And I wish we knew the rules behind this. Is it the gene? Is it the way it's expressed? Is it the timing of expression? Is it just not, you know, CRE-ERT2, we're really not making enough of it, and it's just there isn't enough of it to detect activity. The HAN2, CRE and CRE-ERT work beautifully, and they're both integrations at the 3' end. So I don't have an answer for you. And so we do a lot of work. Ming, it does a tremendous amount of work making alternatives. Yeah. Yeah. Mm-hmm. Yes.
So a very simple technical question.
Sure.
From your slides, it looked like mostly you use mRNA to drive S9.
Mm-hmm.
Did you also try RNP and
Yeah.
protein, and does it?
Mm-hmm. It's a great question. So when we first started doing this, we tried RNP once, and it didn't appear to drive efficient targeted integration. We went back to the RNA, and the idea is that the time that it takes to translate Cas9 is preventing cleavage of the plasmid and inhibiting formation of large concatenamers. Because if we cut the plasmid right away in a test tube, for example, with a Cas9 RNP, it will make linear DNA that's going to link up by non-homologous end joining as a concatemer. But that's all just, we're just, you know, hypothesizing that that's what happens. So there are other groups that use RNP for targeted integration. I'm not saying it doesn't work. I'm just saying that this is what we found worked efficiently for what we're doing. And there are other approaches. You know, we use a vector and short homology arms to drive integration. There are other approaches using linear templates, and they work to varying degrees as well. So, no, it's an important consideration if you're thinking about designing an approach and you want to maximize the efficiency of recovering what you want. Yes.
What type of experiment do you do to confirm that you are in, for example, getting endothelium cells or like mesoendoderm cells?
Yeah, so that's where the switch reporters come in. Based on what we know about HAN2 expression, for example, and all of the lineages that are derived from it, the pattern of switching matches what was predicted. And so for like FLE1B, we know it's expressed in the endothelium of the vasculature. We've done the exact same sort of switch. So with the original CRE lines we made, we did in situ hybridization, and some people still do that too, just to show that CRE is expressed where it should be. I think that those are important controls to do. But the switching, I think, is also valid. So no, it's an important question. Like, how do you show it really is expressed where you think it is? Yeah. In situ.
If I may follow.
Yeah.
I'm wondering if Angel was asking, how do you confirm that these are intact? Those lineages in them. And just those lineages. You know, just by. So is there an independent marker that you use to
Oh, like co-label or, yeah, we haven't done that with this.
Yeah.
Right. We could, in theory, use an antibody. Yeah. To co-label. Yeah. Mm-hmm.
Well,
Yeah.
And following up on that,
Mhm.
notice the expression, maybe the next slide, the HAN2, which is a gene I'm not familiar with.
Mhm.
There's definitely endothelial cell expression going on the front. Is that something that is normally known to be expressed?
That was a surprise. That was a surprise.
Okay.
Yeah. Mhm.
Another question?
Yeah.
I was a little confused about the first of the probably universal guide sequences work. Is that the idea that it includes past management slice up the predators?
Mhm.
It's like,
Mhm.
it's kind of like a past management site.
Yeah, so it's a site where Cas9 will cut on either side of the cargo and liberate it. And once you have the homology arms free, then there's end resection, and you get microhomology mediated end joining or single strand annealing to integrate the cassette into the genome.
So you can inject it as a plasmid, but still have interest.
Right. So the idea is that you have in vivo liberation of the cassette as opposed to doing it in the test tube, which would cause a concatemer to form. Yeah. 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 Yes.
If the core PhD and a switch system is in an older environment, since you're adding that previously, if you wanted to turn something on at 4 days.
Yeah. Great question. And that's exactly the work that we're doing in order to demonstrate that we can use these tools past embryonic development, for sure, especially in adults. We have adults now with different drivers that we've made that have switch reporters in them, and we're asking, can we treat the adult with tamoxifen to switch in the heart, or et cetera. Yeah, great. Yeah. Yes. Thank you.
Have you guys done very much with directly tagging endogenous proteins to the protein localization within cells, like not in tags that don't have a 2A peptide?
Yeah. Some fusions. Yes, we do have those unpublished, and they work really beautifully. Again, when we're injecting in the embryo that was injected, if you can see the pattern of expression of what you predict, that's really a good sign that the integration happened at the site, the intended target site. And so, yes, it does work. If you have a linker to prevent, allow folding, proper folding, but GFP is still fused to it, it works. Yeah, so.
I don't know how many you've done, but are you finding that some of them just are, too low levels to tag to the fluorescence live? Because, for whatever reason, the protein is just, expressed at low levels?
Yeah, so we found that even with just the 2A reporter lines, that some genes you don't see expression because they aren't expressed at a high enough level. And that's also like, sometimes we want to study genes that become elevated in a disease process. They're not highly expressed in the embryo, but in a disease they would be. And so, I think if you can demonstrate after injection by a molecular analysis that there's some indication of on-target integration, we always do this and sacrifice them and raise them up, then you could recover what you want. Celia Shao, she was one of the first adopters of GeneWeld when we were talking about this in the community. And she made a beautiful IRG1 tagged gene line, which becomes elevated when macrophages become activated. So it's like a beautiful marker of inflammation. It's not very high in the embryo, but once there's inflammation, there is. So yeah, so yes, it works. Oh, no, it's you. Yeah.
Okay, sorry, just a kind of a basic CRISPR-Cas9 question. I think one of the fears, I guess, from a clinical perspective, using this to treat human disease is that there are still off-target effects. And so this would be a great model for you to see that you get integration of your cassette in many places. Do you see that, or are you seeing very specific single-site integration, or do you see some off-target effects?
Yeah, we do see off-target effects. So that the frequency that we cite in terms of transmission of precise alleles are animals that are transmitting a single integration at the site of interest. We've done molecular analysis to show that some of these lines have other integrations in the genome, but we don't consider those lines that are precise. Sometimes, yeah, but also sometimes we find that a founder is transmitting more than one allele. So you outcross it, and there's the precise line that's integrating Mendelian, and then there's another integration that doesn't express where it should, and it's probably just somewhere random in the genome. That happens, yeah, yeah.
I think we're gonna thank Maura once again for a wonderful talk.
Thank you.